All right. I think it's time we get going. I imagine some other people are going to be rolling in. Let's see, there looks like 43 participants right now. I think the registration had about 60. So thanks everybody for joining. I think most people at PABC know me. I'm John Culp. Um, I run, I'm on the faculty of the Blumberg Institute, and I run a company called Conifer Point that does software for hit to lead discovery of small molecules and biologics. And CRO work around that. And so Eldari really came out of a discussion I was having with Lou Casa, CEO of our biotech center. And, he was aware that we were doing AI at my company from, making desolvation models, better ranking of ligand protein binding energies, protein motion. And so he was talking to me about, really wanting to see more AI being done at our biotech center and the need that, the companies to be competitive, probably need to start getting on the AI bandwagon. Jess, if you go to the next slide. And I started thinking about that and thinking about this law of diffusion of innovation and really thinking, we have our techies and our visionaries that are the innovators and early adopters. We're probably in that early majority phase of adapting new technologies. But, I was thinking about our biotech center, and there's 50 companies at PABC. There's 30 companies down at the B+ labs in the Sierra Center. And I'm not as familiar with the Sierra labs, but up at PABC, thinking like out of the 50 companies, I'm probably aware of maybe 3 or 4 that are doing work with AI. So that's about 5% of the companies at our biotech center doing AI stuff. And yet, when you think about the AI innovation trend, we're almost in the middle of this bell curve right now. And so I started thinking, yeah, Lou's right. We need to get more AI done. So that was really the inspiration for Eldari Bio, which Eld is sort of fire of life and Ari is sort of the best of thinkers. So it's, our little tagline is innovating life. So with 2 of my employees, next slide, Jess. 2 of my Conifer Point employees, Jess and Luce, we decided to found Eldari Bio, an AI software services company. And coming from the life sciences drug discovery space, really realizing to make this endeavor successful, we really needed to form a larger team. And so we partnered up with Matt and Will. Matt and Will both have experience in applied AI. Matt runs his own software development company outside of the life sciences, was at Microsoft. Will has experience with AI companies, business development, software development. And then Jess's background is in computer science and AI, but came out of a chemistry department at Yale. So she really sort of, bridges our gaps between sort of the technologies and applied AI and the life sciences. And so that's how the team, and we formed Eldari Bio in the 4th quarter of last year, and are kind of, just sort of getting going with our first funding this year. And so that's the story with Eldari Bio. And so what we decided to do today was, when I'm walking around the biotech center, a lot of people say, John, I don't understand this AI stuff, and I think that makes us a little worried to use it when people don't understand it. Now we look at the large language models like ChatGPT and Bard, people are starting to use those. They still say, I don't get how it works. And I know as a scientist, we like to understand how things work. So we're not going to be able to fully explain AI today, but we're hopefully going to get your feet, increase your knowledge, get your feet wet a little bit, and sort of explain, instead of just seeing clouds or little circles with lines between them, start to understand some nodes and layers and like what the AI is actually doing with some case studies in life sciences. So with that, I believe Matt's up next. Thanks, Matt.
Hi, good morning, everyone. Yeah, I'm Matt Kowalczyk. I'm coming to you from the rainy and dark Pacific Northwest over in Seattle. I've been running a software company for 17 years, and for the last 3 years, we've been helping clients specifically in the area of artificial intelligence, and not just theoretical, but applied, and building custom models for their specific business needs. And so when John came to talk to me about how could we apply AI to the biotech world, I had a ton of different ideas. Now, he and Jess are the experts in biotech. I'm just the expert in AI, and I've been building software for 3 decades. So I feel confident that we can come up with a great solution. It's kind of like the peanut butter and chocolate scenario where two great tastes taste great together. We are that peanut butter and chocolate, and we are going to be able to help you and all your needs. So as we get into this, let's talk about AI. We're going to start at the very basics and beginning of AI. Some of this you probably all already know, and that's fine. But I want to give you a groundwork and a foundation to start from so that we are all talking the same language. So with applied AI, I mean, you're seeing it everywhere, every day. And so you can look at things like ChatGPT. I'm sure some of you have an Alexa that you're using somewhere in your household, or a Google, and you have those are voice-enabled artificial intelligence. And then there are very simple things, subtle things like your Photos app. And for example, in both the Google and the Apple Photos app, if you just open it up and say search on the word snow, it will find all the photos that you have in your library that contain snow. How is it doing that? It is using computer vision and recognizing snow in all the photos. And so you're able to play with AI in the everyday things that you're using. So, but that's interesting, but how does that actually work? Let's move to the next slide. So I thought it would be fun if I asked ChatGPT to tell us what is AI. And so instead of me telling you what AI is, I'm going to have an AI tell you what AI is. And so it is a branch of computer science that aims to create machines capable of intelligent behavior, simulating human-like abilities such as learning, reasoning, problem-solving, perception, and language understanding. Wow, that is a word salad. And I'm sure that didn't help explain AI to you at all. And that's part of the issues with AI. But let's get into this a little bit more, and I will demystify it and tell you from the very basics what it is. So let's assume you have some data, and with that data, you are able to create some results. And these results you build manually through iterations and iterations, and you realize this data has some meaning, and you're able to create a result from that. And you do this all mechanically. But with AI, what we aim to do is build a model that takes away that manual process. And so we have your data, you have your result, and then we train this model. It's called a custom model that will go in and be able to do all of that math for you that go from data to manual result. And once you have this model that has been trained, then you can go through and apply the same model to similar types of data. So instead, now you have a new set of data, you use that model you trained in step one, and now you can get a predicted result. So it's data, model, result. And that's the basis of AI. You're taking data, applying this model to it, and then you get a result. Okay, that's great, Matt. What the hell is a model? Okay, you just told me this. Okay, a model is really just a structure with neural networks and nodes and all these different layers of computer science data types. And with that, you have an algorithm. An algorithm is part of this model that teaches the data types and the neural network how to learn. And the more you teach it, the more it learns and the better it becomes at predicting. But there are specific parameters, weights, certain variables that you have to add to the model to teach it to be more interested in one thing versus another thing. And then once you have this whole thing, programmatically, you can access this model with an API that's wrapped around it. And so when you see a model that's used in the real world, it's uploaded to things like AWS SageMaker or Azure, and then your application can access this model and get those predicted results by pushing data to it, and the API will give you a result. It's really that simple. Now let's take a look at some real world examples that are relevant to you where AI has had huge effect in the biotech world. Now, again, disclaimer, I come from the computer science world, not the biotech world. So some of these things I might not get exactly perfect. And I've put citations at the bottom in each one of these case studies for you to explore it on your own. So we will share these slides afterwards and you can take a look at it. So what you're looking here is a protein, a folded protein that was discovered using predictive analysis. It came out of a Google study called AlphaFold. And with this, Google's DeepMind was able to predict all these new protein structures. How did it do that? Well, it trained a neural net on thousands of different types of proteins, created a model, and then was able to predict 214 million unknown protein structures that people hadn't thought of before. Again, data, model, result. And this, with those results, led to all these great new drug discoveries. So that's just one. And this is using a type of AI called predictive analysis. Let's move to the next one. And in this one, you'll see that it is, in this case study, it's about ICAD and doing cancer detection using another type of AI called computer vision. And with computer vision, you essentially feed a neural net lots and lots of photos of cancerous and non-cancerous images of this thing happens to be a mammogram, breast cells. And you train it on what is cancerous and what is not cancerous. You create this model, and then you apply this model to a new set of data. And what they have found with this ICAD cancer detection tool is they've been able to increase the number of positive detections from 3.8 to 6.2 per thousand, and decreasing the false positives by a significant amount. Again, another great leap in applications of how AI can help the biotech world. Now, this is specific around computer vision. I'm going to talk about a third one. And now this one is really fun. And this came out of MIT. And this, some of you may have seen this. This came out last month. There was a big article that was published about antibiotics discoveries for MRSA. Now, this uses a combination of machine learning and game theory. And in this particular study, they used the Monte Carlo tree search. They applied this to an AI model in order to discover these brand new classes of antibiotic candidates. And with that, what they ended up doing was training a model on 39,000 different compounds. And they were able with that to determine what's positive, what's negative, and give this the ground truth, train this model. And with this model, they were able then to take this and apply this to, go ahead, Jess. Apply this model to 12 million other compounds. And with that, they were able to come up with 283 compounds that were potentially viable candidates for antibiotic MRSA. And with that, they were able to come up with 2 out of those 283 that were actually effective against MRSA. And it's the first any antibiotic MRSA compound that's been effective in 38 years. So incredible discovery. Again, I want to stress, we went data, model, result. And those results were game changing. That never would have been able to be figured out without the power of AI and the ability to train these models. Go ahead, Jess.
So I just want to comment, Matt, that was a really impressive study, that MRSA one. It was published in Nature, and those 12 million compounds were commercially available compounds. And yeah, the hit rate, they selected 282, got 2, so it was a little bit less than 1% hit rate. But still, getting 2 active hits with 90% effectiveness on MRSA was very impressive, and it was a really fun study to read about. And with that, we're going to be transitioning over to John.
Yes, go ahead.
Great. Thanks, Matt and John. So like John mentioned, I really exist at the bridge of machine learning and chemistry. I exist there and love explaining machine learning for chemistry and life sciences so much that I actually made a course at Yale for the graduate department in the chemistry department describing how to use this for chemical and life sciences applications. So I'm very happy today to be able to take you guys through a case study where we're going to talk about how you can marry machine learning and life sciences. And my goal with this is really thinking about what are the questions and the structure that people looking to combine these two fields have to think about in order to solve a life sciences problem. We're not going to look too much into the small details around this experiment or the construction of their very specific problem. That's what the citation is for. But with that, let's get into what their problem actually is. They want to look at generating linkers of desired lengths between two fragment molecules that create more potent drugs with desired physical chemical properties. So let's go back to that data model result framework that we've been building up. To solve this problem, they started off with a Kemble-Smiles dataset. So Kemble is a dataset of 2.4 million compounds. And just like if you wanted to teach a high schooler biology, you're not going to throw the entire works of biology publications at a high schooler and expect them to learn biology. No, just like that high schooler, for a model, we need to pare down the information that we are giving to that model in order for them to pull out what information we actually want them to learn. So that's what the scientists in this paper did. They took those 2.4 million Kemble-Smiles and they started by paring them down to drug-like molecules. Then they pulled them down into reaction-specific tuples that had just the fragment molecules that they wanted to build between and potential linkers. They removed duplicate molecules, checked for chemical feasibility. We don't want any Texas carbons. And they removed rare Smiles tokens because they didn't want any cases that they might not be covering in their model very well. And like we've already covered, models are oftentimes discussed as these clouds or black boxes, or if you're lucky, you get a pictographic neural network like I'm showing here at the bottom. But I want to assuage you guys of any fears that we're going to do that. Because really what's going on under the hood of those clouds and black boxes is just statistics and function fitting. We have some output and we have some input. That would be just like your y and x of a basic mathematics function that we learned in our early math classes. And what a neural network or any sort of machine learning model is trying to do is figure out what that m and b of that function are. Now, how do you do that? We're dealing oftentimes in functional spaces that are highly nested, highly nonlinear. And so we use gradient descent methods and other similar optimization approaches in order to determine what those M's and B's are. But in order to do that, we need to make sure that the data we have is as close as we possibly can get it to the X's and Y's being similar to each other. If we make a very complex request of our model, then we're going to have to give it a lot more data, and we're going to have to give it probably a lot larger of a size in order to, for example, jump between a picture of a molecule to what its cytotoxicity might be. Versus if you gave it the molecule in 3D coordinates, information about the partner that it's pairing with, and then ask for the cytotoxicity. It's very important to get those two close. So giving you this high-level overview, I want to take you into what their model in this paper actually looks like. So jumping from gradient descent, y equals mx plus b, their model is an encoder-decoder recurrent neural network utilizing desired feature-specific reinforcement learning. That's a lot of word soup, so let's break it down. A neural network is just a bunch of nested functions. Recurrent neural networks are then neural networks where the data in those functions can filter back into earlier steps of the functions. For example, if there's a part of your function that is extra important and you want to make sure that you spend a lot of time updating those weights, a recurrent neural network is going to help with that. The encoder part is trying to take our human input, which remember might be farther away from our output than we may want, and it gets it into a machine understandable space, much more mathematical than we may be thinking about it in. From that machine mathematical space, the decoder is then going to transform it back into that desired output that we want in the end. Reinforcement learning, which is this bubble we have up here at the top, is then effectively a toddler. It's a model that learns by interacting with its environment. Just like a toddler may be able to crawl and grab things, the reinforcement learning model is going to try different actions, and then based on the responses it gets from its environment, it's going to update its weights in order to fit best to the outputs we're looking for. So what types of results did they end up getting in this case study? Well, they found novel drugs that were similar to drugs with demonstrated improved in vivo efficacy. They found improved docking scores, and they were able to constrain the molecular generation to those molecules that had physical chemical properties that they desired. So with that overview of the case study, let's now get into how we think about breaking down a problem for a given life sciences problem. Again, here is what that problem is. We're generating linkers of desired length between two fragment molecules to create more potent drugs with desired physical chemical properties. So that first part, we're generating linkers. What does your ML scientist friend have to think about first? Well, we have to think about the linkers are going to be our output. That's what you're telling me you want. So what format does that output have to be in? Is it going to be another SMILES string, or do you need it in 3D coordinates? Also, do you want the full compound SMILES string, so an assembled fragment linker compound, or do you just want the linker? Also, you said you want to generate linkers, so you're looking for us to generate new compounds. That means that we need to use generative models, of which there are many different types, that some of which I've listed here. LSTMs, for example, are very good for sequence type data, so probably not what we're looking for in this specific case. So in this case study, they decided their output would be SMILES. They wanted the full compound to be SMILES, and they made a custom model like I already showed you. It's a mix of autoencoder, encoder, decoder technologies, plus reinforcement learning models. Then we have to think about the input. We say that we want it between two fragments. So again, is that input going to come in as SMILES, 3D coordinates? Are we going to tell the model where we want to grow from? In this picture on the side, I have two fragments here, and that star, that red star right there, is indicating where we want to grow from. We also have to think about the encoding format. I mentioned that how the model learns is going to be heavily dependent on how close your inputs and your outputs are. And so the encoding format is exactly that. What type of format are we putting in so that, and what type of information are we putting in so that the model can understand how to translate that information to our output? So you could do a token array, occurrence encoding, hashes, a bunch of different word salad again. But this is something that machine learning scientists have to think about. Then, and perhaps most importantly, and very much married to the encoding format, is how much data you need. So if we have an encoding format that may be more understandable for humans, but is less understandable for the machine, then we're probably going to need more data in order to describe that relationship. Also, how big of a chemical space are we thinking about? If it's just small molecules, that will require a smaller amount of data. If it's proteins and the entirety of chemical space and periodic table, that's going to require a lot more. So in this case, they specifically are using around 10 million small molecule compounds for their data set. And they decide to have SMILES and growth vector input components, and they use a token array for the encoding. Now we get into the restraints. What they want in this problem are making linkers that have a desired length, as well as they have physical chemical properties that give more potent drugs. So ding, 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 a machine learning specialist is going to be thinking, it's a constraint problem, reinforcement learning. So when we start thinking about reinforcement learning, we have to also think about what is our property restrictor going to be? Are we going to be restricting those properties in post-processing? So after data's come out of the model, we say, oh, well, this linker happens to be 6 carbons long, or this one's 5 carbons long. Or do we let the reinforcement learning model learn that process as well? We also have to look at how the reinforcement learning model is learning, which is based on a scoring function. This scoring function can be determined by and learned by the reinforcement learning model, but it can also be provided from some already literature available source, or you can even have an outside automated docking program or some other energy program that can be paired with the reinforcement learning model, that when it knows it needs data, it goes to that oracle and that oracle will give it data. So in this case, they used reinforcement learning gating, which means the model is learning how to restrict it itself. The scoring function has both literature-ready functions that are just immediately fed in, as well as being paired with the docking engine in order to get them docking scores. So bringing that all together, we have our problem and we have our input, which are the fragments, the chemical data, which have been filtered down, and then the scoring functions that are literature ready. And then we also have our outputs, which are going to be these SMILE strings of the fragments plus the linkers already combined. So how did they actually put this together to get some results? So in their first experiment they took a literature-based experiment where people had seen that an initial molecule interacting with a cis 193 amino acid residue inside of a protein had a IC50 of 0.3 micromolar activity. And from that experimental result, they had done scaffold hopping in order to determine that there was an improved molecule that had an IC50 of 0.536 micromolar activity. So what they decided to do in this study was take a look at what the features of this new molecule were that had made it better, and then build that into the reinforcement learning scoring function, as you can see in this case, and use that as the constraints that our model would use to generate new molecules. When they did that, and they docked against the original molecule, which they have over here, and docked that new molecule, they found that the docking score had improved. Now, in this case, it's admittedly only slightly, but they were still able to generate from scratch a brand new molecule that was able to find a better docking score. Moving on to another very similar example, they wanted to retain a lysine 68 hydrogen bonding interaction during the docking step. They wanted to make sure their linker length ratio was greater than 70, and their linker molecular weight they wanted to be less than or equal to 200 Daltons. In this case, again, they found an improved docking score for this other new compound that they were able to generate. And really digging down on their ability to control these physical chemical properties, they did some studies looking at trying to enforce across a different range of different types of properties. So they looked at linker length, they looked at linearity, they looked at the flexibility or rigidity of molecules. And what they were able to show is from baselines, if we look at this leftmost figure here, baseline distribution of linker lengths, if they tried to enforce 4 to 6, they got mostly 4 to 6 length linkers, 7 to 9, 10 to 12, or 13 to 15. They saw the same thing with linearity. They started out with a mixture of linear and cyclic linkers, and when they tried to control for linearity or cyclicity, they managed to enrich only for or mostly for those different types of linkers, and same thing for the flexibility and rigidity. So I hope this case study has given you guys a better idea, not only about the different types of models that may exist or may be being used, but also how we think about breaking down a problem with machine learning in mind for life sciences. Now I'll pass it back to John.
Thanks, Jess. Maybe we can even pause here and address a question that Ian has. You bring up a good point, Ian. You know, bad data in makes for bad models. I mean, we all know that, we're all aware of it, and the variability in some of the life sciences data, yeah, makes it more challenging. I mean, there's always testing and validation we can do, you know, on blinded sets of data, but even if that's kind of bad data, it doesn't work too well. So, you know, data curation, reproducibility of the data are considerations as you're curating the data, and you're trying to mitigate that poor model generation because of the data. I don't know, Jess, if you want to sort of add something there. Yeah, garbage in, garbage out is the term everybody uses, right?
Absolutely. There's actually a lot of very exciting developments that we've seen over the past 10 years in the fields of uncertainty in data. So there are methodologies to make multiple models that take data from different sources. So you'd have source one or source one, and you'd have a model one. You'd have source two of data, and you'd have a model two. And then you have a third model that combines them and determines what data among these different sources is actually reliable. And so you can always do the curation, and if you're in your little bubble, you're not using other people's data, this can work really well. But obviously there's a huge amount of data out there that we should definitely be utilizing if we can. And this combination of models, we often call it multimodal data, because it allows you to bring in not just one type of data. Like you might not just be bringing in cytotoxicity, but maybe you're also bringing in that third that 3D structure as well. So they're coming in different modes. Maybe you have computational and experimental data. You can combine those two. So there are multiple different ways to try to build in ways to have the model figure out certainty. And because the underlying mathematics have heavy roots in statistics, you can actually then tell or learn from the model what their uncertainty on this specific prediction is. So you as a scientist can decide, do I want to trust this or not?
If I could add one additional point to that. And so the importance of data, it's a great question, by the way, is so relevant to any AI engagement. Over the last couple of years, when we've been doing these AI engagements, we have found that 60 to 70% of both the time and expense in any AI engagement is due to the harvesting and the curating of data and putting it in a position so that the models can be trained to get an expected result. And so understanding your data going into this and making sure that it's clean and usable is highly important.
Yeah, thanks, Matt, and thanks, Jess. Yeah, it's really fun to think about those uncertainty models, Jess, and how we might get compounds profiled in cell type A, cell type B. The cells are immortalized differently, have different pathways going on, but then using some of those models to tease out the differences. So thanks for that. So, part of this lecture, we're happy to give you a couple case studies and hopefully increase your understanding of AI. But, we are trying to get Eldari Bio off the ground here and are hoping that if you're interested, you'll contact us and discuss your data, discuss your company, discuss, how technology and AI might improve, your business, help you get more clients, remain competitive. And really, we're hoping to sort of democratize the AI for the Biotech Center. Next slide, Jess. I don't remember if we kept this. Oh, we did. So, we look at AI, and I had my law of diffusion of innovation, and I think we're somewhere in the middle there. But just look at the U.S. government and how much it's increased its spending on artificial intelligence, and they're setting up committees to regulate artificial intelligence. And sort of look at the funding to AI-related startups. It sort of peaked in 21, but that was also due to COVID. If you look at just general VC funding in life sciences, it peaked because people could have so many more meetings in COVID. But we're sort of in a little bit of a VC winter, but we're hoping to come out of it. But if you're going to remain competitive for VC funding, you're probably going to have some AI technology that gives you a competitive edge. As I mentioned, I've been hearing from NIH and DoD reviewers and program managers, almost all of the grant application and contract applications they're getting now have an AI component with them. And so, Eldari's happy to help out, help write that section of the grant, provide that expertise, subcontract on your grants. But even using things like large language models, business, our C-level executives, CEOs, business leaders are increased productivity. When we're writing those significant sections or those commercialization plans, having ChatGPT or Bard provide a lot of good references very quickly. And AI could sort of identify and uncover unique opportunities for your business that you might not have thought of before, and make your employees more productive. So I think there's a lot of reasons to take that jump and get into AI. And I hope I've convinced you that we've got a great, intelligent team here to help you do that. And so, our business model, we're looking at doing proof of concept studies with partners at the Biotech Center for 10 to 30K. I think this is a little bit of a low number, but between two, if you were to hire your own AI full-time FTE, you're probably looking to spend between 200 and 300K per year. And so, anyway, we're offer the support, and we're going out to PABC and Sirius Center because we're here. We're local. We can do face-to-face meetings. I'm there about half-time, maybe more. And so this is how we're going to try to launch the company this year and build up our AI at our Biotech Center.
Hey, John, can I interrupt just 1 second and go back to that previous slide? But I want to explain the engagement model a little bit so you understand the proof of concept. What we're basically doing is we're going in and we're going to understand the efficacy of your data and understand whether or not your data is situated, and can we build a model that would eventually get you to a point to provide desired results. So a POC is going to basically arrange the data that you have in your business, look at it. We're going to try and build a model out of it that'll give us some of the expected results. And if we prove that we can actually do that, then we will do a larger engagement with you where we build this to be part of a vital part of your business. Now, so the important thing here is that you can dip your toe in the water and test whether or not you're ready for AI without spending a lot of money. It's a very small amount in order to understand where you're at in the AI life cycle. And so we can provide that very quickly. And these proof of concepts are a month, maybe 2 at most. And you'll have all the answers you need. And you can change the way you structure your business if you're not ready. But if you are ready, then you could decide to take a bigger bite at the apple. And that's the whole point, is it's a limited risk to assess where you are and if you're ready for artificial intelligence. Thanks.
Yeah, thanks for adding on there, Matt, and clarifying. So here's my email, or one of my many emails, but here's the email you should contact me for Eldari. Just simply john@eldaribio.com. We have our website up and going. It's kind of bare bones, but the company just got started and launched. And I'm hopeful to hear from you. Matt or Jess, do you want to talk about, there's a question in the chat box about data confidentiality when using a tool like ChatGPT?
I'm happy to chat about that. So ChatGPT comes in multiple flavors, and that's a large language model, and that's going to be slightly different than a lot of the other things we will end up doing for you, because we're building custom models that aren't necessarily reliant on a large language model that is provided by OpenAI. OpenAI provides the ChatGPT, for example, and that's based upon GPT-4. However, to directly answer your question, there are 2 main flavors of GPT-4 Turbo. One is available to the public, and that's the free version that you use, and they retrain the model based upon any data you upload to the free version of ChatGPT. If you pay for the enterprise version, like we do, or you access the OpenAI models through Azure, which we also do, we do a lot of that, none of that data is uploaded and used to retrain that large language model in those particular cases. So the privacy is, and that's very important to Microsoft, it's very important to OpenAI, and quite honestly, it's important to us to make sure that we keep your data, and your data is yours and not used to retrain a large language model. Now, however, most of what we've talked about here is not large language models, it's custom models. So the model will be trained from scratch. We might use, like in computer vision, we might use a third-party model and expand upon it, and it's called fine-tuning. But a lot of times we're going to build this model from scratch. So it's not going anywhere. It's just going to stay within your instance of Azure, AWS, local hosting, whatever you're doing. And it's not going to be available for training other models across the internet.
Yep. And again, with the fine tuning of models, we would be taking an instance of that model, so it wouldn't be connected to the larger model. So your data would not end up in someone else's model to advise them how to make your drug.
Great. And probably most of the audience wouldn't know this, but when Matt was talking about using Azure, that's Microsoft's cloud computing platform. All right, let me see, looks like some more questions are coming in. So Stefan wants to know how Eldari Bio is different or compared to these AI companies obtaining the big deals. Like in silico medicines, isomorphic labs, and a few others. Well, I'm not familiar with all of those, but a key difference is our business model. A lot of those, like in silico medicines, do internal drug discovery and are competing with other companies, although they do get licensing deals for technology and drugs from larger pharmaceutical companies. But we are a services business. So we're a software as a services business. We're here to provide you with a service, and that is our main goal. Our main goal isn't to develop our own pipeline, develop our own, medical device or therapeutic. We're here to provide a service for companies and to make them more successful. You know, and Silico Medicines isn't there to make, you know, whoever they got their deal with more successful. They're making, you know, themselves more successful. And so I'd say that's one differentiator. I don't know if Jess or Matt want to weigh in.
Yeah, when John approached me about building this company, it wasn't necessarily, hey, we can make a whole lot of money. Let's go do AI stuff. No, it was built to how can we help the biotech center specifically. So we have established this company, we have established our operating agreements, we have established our methodologies specifically to help you. These other companies weren't designed to help the biotech center specifically. We are. That is what we do. We are essentially, you can think of us almost as an extension of it, but we are built and customized to help your specific needs, not the global needs. And as what John said, we're a services company, and the work that we do at the end of the day is yours. It's not ours, it's yours. We're helping and assisting you.
And one more point to that, we are very customer focused in that we want whatever models we make for you to actually help your businesses. A lot of different organizations out there will make you a model and then they disappear off into the ether. That is not our goal. We want to make sure that we can continue our partnerships in the future. We can evolve the models if you get new data that needs updates to the models, and that at the end of the day, this helps your bottom line. We are absolutely customer obsessed.
One thing I want to also tack on to, thank you, Jess. Is we've already had a handful of conversations with a few companies at the Biotech Center. And those conversations and the exploratory conversations they go very similar to, you tell us about your business, you tell us about your data, we put our minds together in the middle of this meeting, we come up with some very broad ideas on how we think AI can accelerate your business or get you outcomes that you didn't think of in the first place. And then we go through and we're going to create a proposal for you on what a proof of concept would be based upon the strength in the discussion that happened in that introductory exploratory meeting. And so those meetings are half an hour. If they want to go longer, they can, but they're not super intense. They're very high level. Then we do a proposal for you, and then we talk about it. So it's and the whole point of that is a lot of transparency. You understand what you're getting, and you understand how it's going to help your business from the get-go. And if the POC proves to not be vital and not work for you, that's good information to know too. I mean, as scientists, you know that negative outcomes are as important as positive outcomes. And so we are taking that exact same model when we design these proof of concepts for you.
Great. Thanks, Jess and Matt. And yeah, at the end of the day, you're right who made that comment that it's a competitive space. I mean, there's a lot of companies that are going to be fighting for that, whether they're saying we're the best in image recognition and computer vision. But, you know, we're here, we're local, we're flexible. You know, we're keeping up with the latest trends in AI. And, you know, those are just some of the reasons that you should choose to work with us. All right, if there's no more questions, I'd like to thank everybody for attending. This was supposed to be a hybrid event, but we've got, you know, we're going to probably get half a foot of snow here in PA today. So working from home today, but thank you all for your time. I appreciate it, and I hope you learned something about AI. All right.
Thank you all.
All right, bye.